Your brain is, it's an absolute energy hog. I mean, it is constantly firing, constantly processing, and generating massive amounts of toxic neurodegenerative waste every single second.
Right. Things like soluble proteins,
Yeah.
amyloid beta, tau, all that bad stuff.
Exactly. But here is the crazy part. If you look inside the skull, the brain tissue itself completely lacks the traditional lymphatic plumbing that literally the rest of your body relies on to clear out trash.
Yeah, it's a huge paradox.
It really is. It's basically like discovering this bustling, high-end restaurant kitchen that has absolutely no sinks, no drains, but somehow the counters are completely spotless every morning.
That is a great way to put it.
Yeah.
I mean, the human brain figuring out how to not drown in its own toxic byproducts is arguably the ultimate biological mystery.
And we are doing a deep dive into exactly that today, right?
We are. Because for decades, we know that cerebrospinal fluid, or CSF, acted as the main carrier, a sort of biological janitor.
Yeah.
And animal studies had pointed toward a waste clearance network, which you'll sometimes hear called the glymphatic system. But the bottleneck was always our ability to actually watch it work.
Because to see it in rodents, scientists had to, drill literal windows into their skulls.
Yeah, drill holes, inject fluorescent dyes, and observe them under heavy anesthesia. And in humans, the historical method required really invasive spinal injections of tracer dyes.
Which is just wildly problematic from a physics standpoint. The moment you drill a hole in a closed skull or inject liquid into it, you completely alter the local pressure.
Exactly. You ruin the system you're trying to observe.
Right. And the moment you use anesthesia, you alter the heart rate, you alter blood flow. You are artificially disturbing the exact physiological forces you were trying to measure.
Which is why this new study in Nature Neuroscience that we're looking at today is such a massive, massive breakthrough. The researchers actually managed to map the exact mechanisms of human brainwashing in living, awake individuals.
Completely non-invasively, right?
Completely non-invasively. We can now trace the precise biological engines driving this clearance. And perhaps most crucially, we can track exactly how that plumbing fails in neurodegenerative diseases.
Okay, to really grasp how groundbreaking this is, we have to look at the technology that makes it possible. It's a new MRI technique called CSF-STREAM.
Yeah, and to understand why we need a new technique, you have to picture the anatomy. This clearance is strictly localized to what are called perivascular spaces.
Right, the PVS.
Exactly. Picture the blood vessels penetrating deep down into your brain tissue. Hugging the outside of those vessels is a microscopic, fluid-filled sleeve.
Like a jacket around the blood vessel.
Yes, that jacket is the perivascular space, and it is filled with that cerebrospinal fluid. But seeing fluid move through a microscopic sleeve deep inside living tissue requires a level of resolution that standard imaging just cannot achieve.
Because most clinical MRIs at, your local hospital, they operate at 1.5 or 3 Tesla?
Right. But this study utilizes an ultra-high field 7 Tesla MRI. That sheer magnetic power drops the resolution down to 0.45 millimeter isotropic voxels. I mean, it allows us to physically distinguish the tiny sleeve of fluid from the solid brain tissue and the blood vessel it wraps around.
But just having a powerful magnet doesn't solve the contrast problem, right? The brain is super densely packed with solid tissue, flowing blood, and water.
Yeah, it's a very noisy environment.
So I was digging into how the CSF-STREAM algorithm actually isolates the brain fluid, and it uses this thing called a T2 preparation module.
Right, the T2 prep.
Basically, it acts as a selective filter that completely suppresses the magnetic signal originating from the blood and the gray matter. It drops those signals all the way down to the noise floor.
It's genius. It leaves the blood and tissue looking pitch black on the scan, while only the cerebrospinal fluid glows brilliantly.
But a glowing map is just a static picture.
Exactly. And the ultimate goal here is tracking mobility. So to do that, the researchers introduced motion sensitizing gradients.
Okay, break that down for us.
The machine applies a magnetic gradient of exactly 3.5 millimeters per second across 6 different orthogonal directions.
So it's tracking movement in full 3D space. I was actually thinking about this, like setting up a highly calibrated speed trap on a dark highway.
Oh, I like that analogy.
Yeah, like if you take a long exposure photograph of a road at night, the static streetlights stay super sharp, but the moving red taillights, they blur and attenuate, right?
That is exactly how it works. Here, if the glowing brain fluid is perfectly still, the MRI signal stays bright. But if that fluid is moving along the gradient, the signal dims.
And then by calculating exactly how much the signal attenuates across those 6 different directions, the algorithm can mathematically reconstruct the speed and the trajectory of the fluid.
Right. And notice we intentionally use the term mobility rather than just flow.
Why is that?
Because this speed trap is highly sensitive.
Mhm.
It isn't just catching fluid rushing linearly like water out of a garden hose. It captures a highly dynamic environment, slow laminar flow, and even a back and forth rhythmic sloshing motion inside those perivascular sleeves.
Okay, let's just pause for a second and lock this in before we look at the actual map of the brain. I want to do a quick recap for you listening.
Good idea.
Bullet 1. The historical roadblock in understanding brain clearance was that our invasive methods, the dyes and the drills, destroyed the natural pressure systems we needed to study.
Right.
Bullet 2. Now, by combining raw 7 Tesla magnetic power with a T2 preparation module to black out the tissue, and bullet 3, a 3.5 millimeter per second gradient speed trap, we can watch the natural sloshing of cerebrospinal fluid in an awake human being.
It's incredible.
Truly. Ask yourself, how many medical mysteries could we solve if we could just watch the body work without touching it?
Well, when we finally turn this ultra-powerful camera on and map the mobility, the data reveals a really highly active system. We measure normal passive water diffusion in human brain tissue at roughly 1 × 10⁻³ square millimeters per second.
Okay, that's the baseline.
Right. But the cerebrospinal fluid mobility measured in this study ranges from 10 to 50 times higher than that baseline.
So it's literally a rushing river compared to a stagnant pond.
Exactly. But that river doesn't flow at the same speed everywhere. There is a massive regional difference. The fastest speeds are recorded right at the base of the brain, specifically in the subarachnoid space surrounding the middle cerebral artery.
The CES-MCA region.
Right. Down there, the mobility hits 0.041 square millimeters per second.
But then as you move away from those massive arteries at the base of the skull and travel deeper into the brain tissue, into the tiny perivascular sleeves of the basal ganglia, the fluid slows down significantly, doesn't it?
It does. In those deep microscopic channels, the mobility drops to about 0.012 to 0.015 square millimeters per second. So it's roughly 3 times slower than at the major arteries.
No, I was absolutely fascinated by another metric they captured here called fractional anisotropy, or FA.
Yes, the FA's score.
Yeah. For those listening, it's a scale from 0 to 1 that measures how strictly directional the movement is. Like a 0 means the fluid is moving randomly in all directions, kind of like a drop of ink passively expanding in a glass of water. But a score closer to 1 means it is traveling with strict purpose along a single axis. And the FA inside these perivascular spaces was remarkably high.
Which is so important because that high FA proves this is an active biological clearance system, not just passive diffusion. The fluid is being forcefully driven parallel to the blood vessels.
So it's not just leaking out.
No, if it were just leaking or passively diffusing, the FA would be really low. This high directionality indicates there are powerful physiological engines constantly pumping this fluid through the brain.
Which brings up the million-dollar question, right? What are the biological engines keeping this fluid moving?
Well, the researchers mapped out 3 distinct drivers.
And to help you remember them, think of the mnemonic HBV, heartbeat, breathing, and vasomotion.
Right. And proving that heartbeat and breathing drive the fluid required a brilliant piece of computational sorting known as retrospective binning.
Yeah, explain how they did that, because the setup is wild.
So during the 40-minute MRI scan, the subjects were equipped with a pulse oximeter on their finger and a respiratory belt around their chest. The researchers recorded every single cardiac pulse and every single inhale and exhale.
Then the algorithm took that massive block of MRI data and retrospectively sorted it. It essentially said, okay, group all the fluid movement that happened a millisecond after a heartbeat into one bin, and group all the movement that happened a millisecond after an exhale into another bin.
Exactly.
Yep.
And to validate this, they also ran a control where they sorted the data entirely at random.
And what happened?
The random sort produced nothing but jagged, noisy lines. But when binned by the cardiac and respiratory phases, perfect, coherent oscillatory waves emerged. The fluid was undeniably pulsing in time with the heart and the lungs.
That is just wow. Okay, so looking at the cardiac impact, the HBV mnemonic, the heartbeat really dominates the large spaces at the base of the brain, right?
Yeah, when you look at the subarachnoid space, or the fourth ventricle, the cardiac cycle creates massive oscillations. And the physics behind this makes total sense when you consider the skull as a rigid, completely closed box.
Right. During systole, your heart forcefully ejects blood into the arterial system. When that surge of blood reaches your brain, the entire vascular network expands.
And because the skull cannot expand to accommodate that extra volume.
The brain tissue physically squeezes the cerebrospinal fluid. It forces it downward out of the skull and into the spinal column.
Exactly. And then during diastole, as the heart relaxes and blood pressure drops, the fluid rushes back in. The heart is effectively treating your brain like a giant mechanical bellows.
I love that visual, but that violent mechanical pumping is naturally dampened as you move deeper into the brain tissue, right? Because the vessels narrow and the kinetic energy of the pulse dissipates.
Right, which is where the respiratory impact, the B in our mnemonic, steps in. Deep inside the brain, in those microscopic perivascular spaces, breathing and heartbeat actually share the workload equally.
Really? 50-50?
Basically, yeah. The amplitude of fluid displacement driven by the heart in those deep spaces was measured at about 2.5 to 2.8%. Respiration drove a nearly identical 2.4 to 2.6% change.
Wow. So how does breathing actually move fluid in your head?
It's all about pressure. The changing pressure in your chest cavity every time you inhale and exhale draws venous blood out of the brain, and that creates a slight vacuum effect that pulls the cerebrospinal fluid along with it.
So deep in the tissue, these two distinct rhythms, the pulse and the breath, create a vital mixing effect, constantly churning the fluid to ensure stagnant waste doesn't settle.
Exactly. It churns the waste.
Okay, time for our second mini recap. Let's synthesize what we know so far.
Go for it.
Bullet 1, the fluid in your brain is moving 10 to 50 times faster than normal water diffusion, and it's traveling with strict directionality along the blood vessels.
Yep.
Bullet 2, at the base of your skull, your heartbeat is the primary engine, violently squeezing fluid out as the arterial network expands.
Right.
And bullet 3, deep in your gray matter, your heartbeat and your breathing act as dual engines, churning and mixing the fluid 50-50. I mean, have you ever considered that the depth and rhythm of your breathing is literally washing the deepest corners of your brain?
It's pretty mind-blowing. But that still leaves the final hidden engine of clearance.
The V in our mnemonic. Vasomotion.
Vasomotion. And this mechanism is profoundly important because it connects the mechanical plumbing of the brain directly to why humans require deep sleep.
Okay, so what exactly is vasomotion?
Vasomotion is the spontaneous, low-frequency oscillation of the blood vessel walls. Completely independent of your pulse or your breathing rate, the smooth muscle cells lining your cerebral arteries just naturally contract and dilate.
Right, this inherent twitching happens at a frequency of about 0.1 hertz, which means the vessel expands and contracts once every 10 seconds.
Yeah, but observing this experimentally presented a massive hurdle for the research team.
Because you can't just put a sensor on it.
Exactly. You can track a pulse with a finger clip, you can track breathing with a chest belt, but there is no wearable sensor that tracks the spontaneous twitching of deep brain arteries for that retrospective binning algorithm.
So since they couldn't passively track it, they decided to forcefully induce it using a visual stimulation experiment.
Yeah, this was so clever. While the subjects were inside the MRI, they were exposed to a flashing checkerboard screen. The checkerboard flashed at 8 hertz for exactly 5 seconds. Followed by 5 seconds of a blank gray screen.
And the timing is the genius part there. 5 seconds on, 5 seconds off. That creates a 10-second cycle, which is exactly 0.1 hertz.
Right. By forcing the visual cortex to process information on that strict 10-second loop, they force the local blood vessels supplying that area to dilate and contract at the exact natural frequency of vasomotion.
And the results totally confirmed the hypothesis.
They did. The localized cerebrospinal fluid mobility in the visual cortex increased by an average of 1.2%.
And they simultaneously tracked the BOL-D signal, right? The blood oxygenation level dependent signal, which is like the standard measure of blood flow.
Yeah, and the magnitude of that blood flow response correlated directly with the increase in fluid mobility. The physical expansion and contraction of the vessel walls was directly sloshing the perivascular fluid forward.
Now, obviously, we do not spend our nights staring at flashing checkerboards.
Thankfully.
Right. But here is a biological punchline. During deep, non-REM sleep, your brain naturally generates massive synchronized electrophysiological slow waves. And guess what the frequency of those sleep waves is?
0.1 hertz.
Exactly, 0.1 hertz.
So that localized visual experiment serves as a perfect model for what happens globally every single night. During deep sleep, those slow electrical waves entrain vasomotion across the entire brain.
So blood vessels pulse in massive synchronized waves.
Yes, acting as a unified pump to forcefully wash away the toxic proteins accumulated during waking hours.
So we have our pristine, healthy plumbing system. heartbeat, breathing, and vasomotion HBV all acting in concert to drive fluid deep into the perivascular spaces. But the true value of CSF-STREAM is seeing what happens when those pipes break.
And that's where the clinical side of the study comes in. They brought in 8 patients suffering from cerebral amyloid angiopathy, or CAA, and matched them against 8 healthy controls.
CAA is a devastating, incredibly common neurodegenerative disease in older adults, right? Heavily intertwined with Alzheimer's.
Very much so. The hallmark of CAA is that toxic amyloid beta proteins don't just float in the tissue. They aggressively accumulate inside the walls of the blood vessels themselves.
Oh, wow.
Yeah, the vessels lose their smooth muscle cells, becoming heavily calcified, rigid, and really prone to microscopic ruptures.
So when they scanned these CAA patients, the data initially looked like a paradox. Because remember how the fluid highway at the base of the brain, the subarachnoid space, had the fastest mobility in healthy people?
Right.
In the patients with severe CAA, the fluid in that exact area was moving 20% faster.
Which is wild because a 20% increase in speed sounds like enhanced clearance, which makes no sense for a disease defined by waste accumulation.
Right, it seems backwards.
But the second metric, the fractional anisotropy, or FA, revealed the truth. While the fluid was moving 20% faster, its FA was 10% lower.
Meaning the flow was significantly more chaotic, splashing randomly rather than moving with strict parallel purpose.
Exactly.
I was thinking about it like watering a dead potted plant. You know, when the soil in a pot is healthy and aerated, the water gently soaks down deep into the roots.
Yeah.
But if you neglect that plant for months, the soil becomes severely dehydrated, rock hard, and completely stiff. When you finally pour a glass of water into the pot, it doesn't penetrate the soil at all. It just pools on the surface and violently, chaotically rushes down the gaps between the soil and the plastic pot, entirely bypassing the roots.
That is the exact mechanism happening in the brain of a CAA patient. The amyloid buildup destroys the elasticity of the vessels and turns the surrounding tissue stiff and unyielding.
So the fluid just can't get in.
Right. The cerebrospinal fluid can no longer penetrate the deep microscopic perivascular channels. Because it faces immense resistance pushing into the deep gray matter, the fluid simply bypasses the brain entirely. It rushes rapidly and chaotically over the surface spaces instead.
So the high speed on the surface is really just a tragic illusion of health. It is actually the ultimate symptom of profound structural failure deep inside.
Yes, and the spatial resolution of the 7 Tesla MRI mapped this perfectly. This faster, chaotic flow was strictly confined to a 1.7 millimeter radius surrounding the large surface arteries.
And what about beyond that?
Beyond that 1.7 millimeter boundary, down in the deep tissue where the clearance is actually needed, the fluid was just stagnant. The perivascular spaces were swollen and enlarged, but the mobility was dead.
Wow. The implications of visualizing this bypass mechanism in real time are just staggering for clinical medicine. I mean, historically, evaluating a new Alzheimer's or CAA drug meant waiting years to observe a patient's cognitive decline.
Right. But with CSF-STREAM, a patient could be administered a vascular elasticity drug, and a month later, we could just scan them to see if the fluid mobility deep in their basal ganglia has actually improved.
We're looking at the ability to map the mechanical onset of neurodegeneration years before the first symptom of memory loss ever appears.
Exactly.
We have dissected an incredible amount of biology today. We transitioned from relying on invasive dyes and drilled skulls to tracking the exact trajectory of human brain clearance using the raw 7 Tesla power of CSF-STREAM.
A huge leap forward.
We established that our brain relies on 3 distinct engines: the heartbeat squeezing the massive surface arteries, breathing creating a 50-50 mixing effect in the deep tissue, and vasomotion leveraging the 0.1 hertz slow waves of deep sleep to pump the microscopic vessels.
Or HBV.
Yep. And we saw how diseases like CAA calcify this system, turning the brain into that dehydrated potted plant where fluid violently bypasses the toxic tissue entirely.
You know, understanding that clearance relies on the intricate, shared workload of our cardiovascular, respiratory, and sleep systems completely reframes how we view brain health. It is not an isolated organ. Its daily survival is mechanically tethered to the rhythm of our lungs and the elasticity of our arteries.
Which leaves you with a final, profound question to consider as you go about your day. If the nightly deep cleaning cycle of your brain relies so heavily on the mechanical, rhythmic expansion of your lungs and the steady pulse of your heart, What are the long-term, decades-long consequences of chronic stress?
That's a scary thought.
Right. What happens to the stagnant, toxic waste deep in your gray matter if you suffer from sleep apnea, depriving your brain of those zero point 0.1 hertz slow waves? Or if you spend your entire workday taking shallow, anxious breaths?
It really makes you wonder.
Could the physical depth of the breath you take today dictate the amyloid plaque building up in your brain 20 years from now? Something to think about the next time you take a slow, deep breath. You might just be giving your brain the exact mechanical push it needs to take out the trash.